Hello, my name is Ian Hoffecker, and welcome to another video on machine learning in biotechnology. And in this video, I'm going to be talking about two topics, classifiers and optimization, and how they're unified by the concept of Gaussian mixture models, which is a form of unsupervised machine learning for clustering structures found in data. So first, a bit of review from the last lecture. So we talked about this idea of the Monte Carlo method, which allowed you to start with some probability distribution function, or alternatively, some process or programmable series of steps that lead to branching outcomes. And either of these processes would give you the ability to produce randomly sampled data. So in the case of the probability distribution route, you have a known set of parameters. You would compute a cumulative distribution function, a CDF, and for every y value on that CDF, you have an associated probability that monotonically maps to a unique x position. So this allowed you to sample from this distribution by picking random numbers uniformly between 0 and 1 associated with these probabilities that then get remapped to the x values. And then this allowed you to simulate data, or alternatively, if you could create these branching processes programmatically through an understanding of the underlying process, even without knowledge of an analytical distribution, this would also allow you to simulate data and produce actually the frequency distributions that would otherwise require direct knowledge of the analytical function, sometimes which might be quite hard to know. And so that was the first step, the idea that you can generate data using this method. And this is also sort of an analogy to what's going on when physical data is collected, that you have some underlying generating process. And we could also use this data as the starting point to go backwards, go the other way with the maximum likelihood estimation method. And the idea here is now we're starting with a distribution on this end. We know that there's some underlying probability distribution governing this process that's responsible for generating this data, and it has some set of parameters. And so what we might do is hypothesize a distribution function that explains this data, and then systematically change the parameters, theta here for parameters, and check the probability of each data point in our data set given that set of parameters, what probability is associated with that for this particular model, this particular distribution. And this was called the likelihood. And the idea was that if you check several different parameter values, so here we have parameters being varied on the x-axis, and we're checking likelihood values here, and that there's going to exist some point, a maximum of this function, which is going to correspond to the best set of parameters, i.e. the parameters that maximize the likelihood that you're going to observe the data. And this would allow you to then fit the distribution to your data. So we would have a resulting analytical function from our hypothesized distribution here, as well as a set of parameters that would hopefully dovetail nicely with the data that we have. So that was the basic idea. And the workhorse math behind this was the maximum likelihood estimation method, which relied on this likelihood function here, where we're computing the overall likelihood through the product of individual likelihoods, so one for each data point, or probability for each data point. And we're taking the log of this in order to turn this into a summation instead of a product. And the idea is that we want some function that's going to change the parameters and find the best set of parameters that maximizes this particular value here. And then we said that because of historical reasons in machine learning, you're typically minimizing something instead of maximizing something. And so by taking the negative of this log likelihood, we are then able to minimize this function. So then the negative log likelihood becomes the function that we're trying to minimize by changing these parameters. And we could see that by sweeping through different parameters here of mean and standard deviation, we would get different values of this likelihood function. And in this kind of grid-like search, we would identify more and less promising candidates, with the most promising candidates having a high peak value here. But the kind of key thing that we didn't really discuss last time was this idea of the minimization or optimization, that when we did this in practice, we just kind of ran this function and it went on its own and performed this minimization or maximization step for us. And so that's what I want to talk a little bit about today, this idea that we are relying on computers to perform this optimization process. And the idea is we have some kind of function, right? In our case, the negative log likelihood function, and we're trying to find a minimum. And we may not know the shape of this function. And so one kind of vocabulary here is the idea of a global minimum versus a local minimum. And so if you have a local minimum, it means that the places locally close to that function are going to go up, meaning that if we had an algorithm that was just going to try to get lower and lower and lower values of this loss function, it would potentially get stuck in one of these local minima, because if we're starting, for example, on this point in the hill, the way to get lower values in the function is to proceed to the right here towards the local minimum, whereas a global picture that we have would tell us that no, in fact, you should actually go up over this hill here to find the global minimum. And so another idea is in optimization problems is the idea of constraints, or this conditional optimization. And so the idea here is that while you might not have a unique global optimum in your function necessarily, you might have a particular window where that is the case, and that window might be the area that you're most interested in. So you can see that if we slide this window around, here we do have a global maximum in this case. If we slide this window a bit to the right, now there's two. So there's no longer a unique one. And if we, in fact, open this up to be unconstrained, in fact, this function, maybe it goes off forever to infinity and minus infinity. And in fact, there isn't any global maximum. So constraints can be very important for finding your value, parameter value that you're interested in. And so the technique that, so we're just going to talk about this at a conceptual level, but the technique that people are using to find these minima is something called gradient descent. And this is based on a lot of calculus, because the idea is that we're going to be looking at the rate of change of our function. And so an analogy to this might be kind of picturing what it would be like on a hilly landscape with your eyes closed. So if you have no notion of where the global minimum happens to be, but you have the ability to take steps and determine whether those steps are going down and approximately how fast they're going down and in which direction they're going down the most. And so that's the idea behind gradient descent methods, that you sample a few values wherever you happen to start, and then you're computing the fastest route down the hill in that local patch. And so we have this idea of the step size here. And in machine learning, this is often referred to as the learning rate. And sometimes this is fixed. So you can have a situation where you have a constant step size, and this can sometimes work, but it can also have problems. So for example, if your step size is too small, maybe you're trying to get down a hill in a mountainside, and your step size is the size of an ant's step size. And if that's the case, it's going to take you a lot of steps to find the global minimum. So you'll often have adaptive step sizes. And if the step sizes are changing, this is sometimes referred to as the learning rate schedule. And so in the last exercise that we did, we just called these functions. So in Python, it was minimize. And in R here, it was this optim function. And we called this method LBFGS, and this is a commonly used variant of gradient descent that is used for a broad range of optimization problems. And so another kind of key aspect of gradient descent is that you're going to be relying on the information about your landscape from your local perspective. So that's kind of the key requirement for these techniques to work. And so they can actually break down when your landscape is bumpy or choppy. And when the information about what's downhill and uphill that you're getting from kind of sampling locally your function does not actually give you accurate information about the global landscape. And so that's happening here in this case with this V-shaped function, where if we look at the derivative, the rate of change, it's constant until suddenly there's a step change here. And then so if we're sampling the rate of change over here locally, we cannot see what's going to happen here. And you can imagine that this sudden change comes in an unpredictable way. And so this could mean that you're searching fruitlessly through this landscape for things that you cannot see. And so if we kind of go back and just sort of review what we did again. So we had this function NLL here in R and here in Python, and it had these two input variables, params and data. And the data here was actually a fixed variable. So we call these args. These are the variables that are fed into the function and are not allowed to change. So when we call this minimize function, we fed in the name of the function, an initial guess, and these args. So these were data points that are needed to compute the probabilities, of course, right? For each probability, we're checking a particular position on our random variable x-axis or k-axis here. But what we do want to change are the parameters. And in that case, it was R and P. So we were interested in these Pascal distribution or negative binomial distribution parameters, R and P. And so these were the ones that are allowed to change. And in the syntax of this minimization function here in Python, the first variable is the one that's permitted to change. And so in the function, we had to feed in a single variable here and then pull those two variables out was kind of how this was structured. So the resulting output would be the final parameter values, and you'd get this complex object from this minimize that we called result. And this would also have, for example, the final value of the loss function, which is hopefully very low. But you can imagine, doing this manually. So here I have an example of how, we could just list our own parameters here. You could do this in an Excel spreadsheet. We could start from some initial values, compute the probability for each data point, and for these parameters R and P, compute the negative log likelihood, get some value, and make a change, do the same thing, and then see if it went down or up. And we could imagine just kind of repeating this calculation manually, tweaking the values of R and P to really get the lowest value we can of NLL, negative log likelihood. And so that's what this function is doing, but much faster, of course. So kind of changing gears here. So in this video, I want to talk about the idea of classifiers. And classifiers are, of course, used in a lot of machine learning applications. So here's one kind of biotechnology related application, which is base calling in DNA sequencing. So this is taken from the Oxford Nanopore website. So the idea here is that you have an electrical signal that's coming from your device, and the different values of this electrical trace here correspond to different bases in the DNA sequence. And so they built a classifying machine learning algorithm that takes the signal and classifies different parts of the signal as different bases, A, T, G, or C. And so this is based on a neural network, a recurrent neural network, but then you have other classification algorithms out there. So a common example in single cell sequencing are clustering methods. These are often used to identify cell types based on their gene expression profiles. And you can also have classifiers for image analysis. So this is an example of DNA nanostructures whose shape is being used to classify the different structures according to their particular states. And what we want to do is I want to talk about a particular type of classifier, which is a Gaussian mixture model. And the reason I want to talk about this is because it's going to allow us to explore a little bit more this topic of optimizers, and also to reuse some of our knowledge from the previous lecture on maximum likelihood estimation. But there's first a little bit of background that we're going to need to discuss this topic. So the Gaussian mixture model is based on the Gaussian distribution. And so we kind of very briefly talked about this last time, but of course, this is a very familiar distribution to most people, also the normal distribution. And the idea is you have two parameters, a mean and a variance, or a mean and a standard deviation. And it's going to give you this bell-shaped curve here centered around wherever the mean is. And the width of this distribution is going to be determined by the variance or standard deviation parameter. And so this is the PDF, the probability density function, the probability function that's describing this distribution here. So if you have a particular X value that you're interested in, this will give you the probability density at that value of X, given that you know some of the values of these two parameters here. And so we didn't talk about this last time, but there is in fact also a generating process behind the Gaussian. And so similar to the binomial process, you know, we did coin flips to kind of show intuitively how the distributions behind the binomial emerge. And likewise, we can kind of do the same thing for the Gaussian. And so Gaussians emerge whenever you have a sum of independent random variables. So what do I mean by that? So if we were to just iteratively sample random variables like this, we would get something like this, a histogram between -1 and 1 here. So we're sampling between -1 and 1. And you can see it's uniform. So if we do this enough times, 100,000 times, you can see we get a completely flat spread of different values between -1 and 1. But now suddenly, over here, let's say we do this 3 times. So we have 3 of these randomly, uniformly sampled variables between -1 and 1. And what we're doing is adding them together. And then that sum becomes the data that we're interested in that case. And even though there's just 3 of these added together, you can already see the Gaussian emerging. So the generating process behind the Gaussian is a sum of independent variables. And so you can imagine why this might be kind of applicable in a lot of situations. So one situation is error, for example. Error emerges whenever you have the cumulative effect of many different variables acting on each other. So let's say we're measuring the length of this bolt here, and there's lots of little tiny unrelated factors that are going to reduce our precision or our ability to measure this thing atomically accurately. Maybe it's the ruler, the angle of our eye, lots of small things that are unrelated. And so they're kind of contributing in one direction or another to the final result. And so they're kind of diffusing our measurement, you could say. And this is analogous to this summing of independent variables. And this is something called the central limit theorem that basically says this happens whenever you do this. You sum independent variables, you build a Gaussian. So that's a single Gaussian, but what we are interested in today is the Gaussian mixture model. And so the idea here is that we actually have multiple Gaussians, and they're going to be mixed with each other, like I've drawn here. And this is actually a type of unsupervised machine learning. And the idea here is that we're going to use our ability to fit distributions to data in order to identify clusters or identify particular subgroups of our data that we're interested in. And so there's 3 kind of components that we need. So 2 of them are familiar. So they're the parameters of each Gaussian. So each Gaussian has a mean and a variance, or if it's a multi-dimensional Gaussian, a covariance matrix. And the third thing that is new is a weight. So how big or how much data is coming from these different Gaussians. So here I've drawn relative weights, and we denote these with the Greek letter pi. And there's one big assumption that goes into this model, which is that we know how many Gaussians there are going to be. And so we're not going to start just yet, but I'm going to give an idea about what's coming. So the exercise for today is to fit a GMM Gaussian mixture model to some simulated cell expression data. So we have, let's say we have this mixed population of cells, 3 cell types. And what we're looking at are kind of observable that we have data from is the expression level of a particular gene. that all three of these cells happen to express, but they express them in different levels. But they don't express them in different levels so completely that their expression levels can be completely separated. And so you can see there's overlap in the distributions of expression levels from each cell. And so when we sequence these cells, each cell gets sequenced individually, but we don't get to know its identity. All we get to know is its gene expression level. And so all we see is this data here, the mixed data. And so the goal of the Gaussian mixture model will be to figure out these three parameters so that we can see what distributions are making up or underlying this observable distribution that we see. So it might be kind of like this. So we have, say, Gaussian 1, Gaussian 2. And if we had access to the ground truth, we would see that there are two unrelated distributions here. But in practice, what you're going to see is the mixture of these two, with these overlapping parts here kind of summed on top of each other or mixed. And so this is the formula for the Gaussian mixture model. So we have a likelihood expression here. So this is going to be a familiar format for a lot of machine learning models. So we're interested in the probability of some data variable premised on a certain set of parameters. And we want to maximize this likelihood basically by changing these parameters. And in this case, it's going to be the sum of k Gaussians. So k equals 3 in this drawing here. And each of those is going to have a weight. So this is just going to be a fraction here, a number between 0 and 1 associated with each Gaussian. So maybe this guy here is 20%, and this one here is 35%, and this one here is 45%. Should sum to 1. And then the second component here is the Gaussian PDF. So if we remember back to the other slide, this is the Gaussian PDF, right? It takes parameters mu and sigma and also x. So we have going to do this for each x variable. And what we can do is the same thing as we did in the last video, where we computed or turned this into an expression that can be dealt with by the minimizer. And so for that, we want the negative log likelihood. The idea is we have this likelihood expression, this one here, but we're going to take the log of it, first of all, the negative log of it, so that we can minimize it. And then our minimizer is going to perform this operation here, find the minimum by changing theta, and then remember that theta, or remember those parameters that gave you that minimum. So our goal then is going to be finding these parameters. And if you do that, you'll get this nice fit here. So this is the mixed Gaussian model, and overlaid on top of the data. And then once we've done that, we can essentially turn this thing into a classifying machine, right? So for each data point, we know whether it's in a region that's dominated by Gaussian 1 in this case, or dominated by Gaussian 2 in this case. And so we could make a call just simply based on, which is more probable. So to the left of this line, Gaussian 1 is more probable. To the right of this line, Gaussian 2 is more probable. So we might just say anyone occurring to the left here, we're going to call it, that's Gaussian 1, like that. And if we had access to the ground truth, we would be able to build this kind of confusion matrix to test whether we're making these calls correctly. So if it's actually 0 and we said it's 0, Gaussian 0, then we could put it in this box. If it's actually 1 and we said it's 1, then we could put it in this box. And then if we made a mistake, actually, if it's actually 1 and it's 0, or it's actually 0 and we said 1, then we put them in these confused boxes here. But we have some more information, right? So we can make these calls, but we also, after fitting the model, we have access to the probability associated with each Gaussian. So for some of these intermediate ones here that are kind of shaded in an intermediate color between green and red here, what we've done is computed the probability that our assignment is correct. And the way you can do that is by computing what's called the odds ratio. So we're going to compare probability densities. So let's say we have a point, x sub i, and it shares probability density values from two different distributions. And in one of those, its value is 0.2, and in the other, its value is 0.3. So when we were doing our classifying calling exercise, we would have picked this one here. This is the more probable one. But we can say how certain we are about that. So if we take, we have the ratio here, it's 0.2 to 0.3. And what we can do is compute 0.3 divided by the sum of these values, 0.2 plus 0.3. So normalizing it based on the sum of these values. In other words, and we get a probability of 0.6. So we call it, the second Gaussian here, but we're only 60% sure about that. And so now we go back to the exercise, and the goal here is to apply this Gaussian mixture model. And the first step here is going to be to transform the data into a logarithm. And so the data, so this is kind of simulating realistic expression data, where actually the values tend to be log normally distributed. So they're kind of, they tend to spread out the larger the values are, and they tend to bunch together the smaller the values are. And so this doesn't look like a mixture of Gaussians at all if we just kind of look at this raw data here. But after taking the log, so we take log base 10 of this data here, and then put it into a histogram again, and now we have this log 10 expression level, which does indeed look like a mixture of 3 Gaussians. And so we might imagine in our sample, we happen to know there are 3 cell types. And so this means that we're going to use this knowledge to build our underlying assumption that there are 3 Gaussians making up this distribution. In the second step, what we want to do is build our mixture model. And so we have this function here, mixture model. And the idea here is that we want to do this part. So we want to be able to compute the probability for each data point. So we're feeding a single data point into this function, and we're also feeding a set of parameters into this function. And so those parameters include the weights, the pi's, the means, these mu's, and the standard deviations, the sigmas. And if we have all those values plus a data point, then we can evaluate this expression here. And so we should get this fitted result at the end. In Python, if we look at the output of this result from the minimizer, we can see the final value of the negative log likelihood function, 2,613. So hopefully this is a low value. I think it is. And we also get x here is the variable that's allowed to change in the syntax of this minimizer function. And so in that case, for us, that was our list of parameters. So these are the things that are allowed to shift. And so these are the final values. So 0.3, 0.3, 0.4, 0.3, a mean at 2.6, for example. So these were the values that it found. You can see it took 75 iterations, 813 function evaluations. And so that's how it's done in practice. And something that you may or may not have encountered is, so if we were to generate some other distributions here, some may be easy, but even if they're easy, they might not necessarily have a unique solution. So if we have these 2 symmetric Gaussians here, and we actually plot the parameter space here as a contour plot, so we're looking at the negative log likelihood values here. So higher is worse, of course, and we're trying to find these minima. And you can see actually that there are 2 of them in this case. So we could either put one Gaussian here, or we could put that same Gaussian here, and it's symmetric because they're kind of the same. And so this means they're actually not a global minimum for this problem, and it's not a convex problem either. And you can also see that we have the issue of local minima in this case here. So we trying to fit 3 Gaussians to this data set, and we can see that there are some local minima that we might get stuck in, which would not be an optimal solution relative to these deeper pockets down here. So that's the minimization step. And then the next step that we're interested in is building some kind of classifier. from this. So what we can do is assemble a table here of likelihoods or probabilities. And the idea here is that now that we've gotten our parameters, so we have, say, 3 Gaussians, we know the means and the variances and everything for these different Gaussians. And what we want to do is, for each data point, compute the probability associated with each of those 3 Gaussian distributions. So we can do that one by one. We go through each Gaussian and go through each data point and spit out a value. And one of those is going to be higher than the others. So let's say, we do this for the first data point, and Gaussian 1 gives us a likelihood of 0.99, whereas the other 2 give 0.005, 0.00499. And then we might say, okay, we're going to assign this data point to Gaussian 1. So this is cell type 1, in other words, based on its expression level. So maybe we can imagine that that could be like a point, right here, deep in the green. It has a really low probability likelihood associated with these other 2 sets of parameters. So that's classifying, and in general, something that is going to arise pretty easily here is challenges with finding a good fit. And you can see this here. The moment we start kind of changing the distribution to be a bit more tricky, we'll get these kind of non-optimal fits. If we fiddle with things enough, maybe we're lucky and we get a good fit here. And so one thing that we didn't cover is actually the kind of full version of this method. So what we've done is a simplified version in order to practice MLE and look at optimization. But in practice, this GMMs, they're fitted using something called the expectation maximization algorithm. And this is because of this high level complexity. You have a lot of parameters and this kind of non-convex landscape in the log likelihood function tends to be difficult for direct use of maximum likelihood estimation computationally. So for more simple problems like the one we did, it's okay. But for multi-dimensional problems, so for example, if we have a 2-dimensional Gaussian here, we may run into some trouble finding a good set of parameters. So when you actually call, for example, the Gaussian mixture function from the scikit-learn Python package, it's going to be implementing this EM algorithm instead. So that's what we used here to generate this 2D GMM. Okay, so that's it for this session. And to kind of summarize, today we looked at probabilistic models and how we can use those to describe mixtures of data or data coming from mixture of sources. We're going to be doing this by fitting multiple 2 or more Gaussians to this mixture of data. And then we talked about optimization algorithms, and these are going to help us find these parameters. I'm going to be doing this using this gradient descent method, where we're looking at some kind of loss function, which is telling us basically how good our parameter assignment is at that particular point. And then finally, we use this Gaussian mixture model, and this actually became a kind of clustering method or unsupervised classifier that we use to separate our data into different categories. And so this hopefully covered some important themes in machine learning, and these ideas should come back multiple times. So we'll continue using optimization algorithms. We're going to continue seeing probabilistic models and maximum likelihood estimation. So that's it for this, and I'll see you in the next one.